Dear viewers, I welcome you all to the session on amino acids and peptides, properties and structure. Starting with introduction. It has been stated that nature is bestowed with more than 700 amino acids and most of them are alpha amino acids. These are cohesively provided by bacteria, fungi and algae and other plants which exist either in the free form or bound up into larger molecules. As ingredients of peptides, proteins and other types of amide and of alkylated and esterified structures. The 20 amino acids, which actually are 19 alpha amino acids and 1 alpha amino acid, are entrusted with the key responsibility of being utilized in living cells for protein synthesis under the control of genes. Therefore, they are a unique category since they are fundamental to all life forms as building blocks for peptides and proteins. However, it is unknown as to why all the other natural amino acids are located where they are, and it is an area of much conjecture. For example, some atypical amino acids are present in many seeds and are not needed by the grown up plant. They dissuade predators through their toxic or otherwise unpleasant characteristics and in this way are thought to safeguard the existence of the plant and thereby contribute to the overall existence of the species of the plant. Peptides and proteins are very vital to the existence of any living form and play a wide variety of roles in living organisms and exhibit a range of properties which varies from the potent hormonal activity of some small peptides to the structural support and protection for the organism shown by insoluble proteins. In this chapter, we will focus on the basic nature and structure of amino acids and peptides. We will try and understand their properties and will also visit the general nomenclature followed for denoting them. Definitions and common amino acids. The very name amino acids are derived from the fact that they have both amino and acid groups in the same molecule. The biologically important ones are alpha amino acids, that is, both the groups which are functional are bonded to the same carbon. The general structure is as shown here on the screen. The sign star indicates that when R is not a hydrogen atom, the molecule is chiral. Amino acids exist in either D or L enantiomorphs or as stereoisomers. The D and L refer to the absolute conformation of optically active compounds. With the exception of glycine, all other amino acids are mirror images that cannot be superimposed. Most of the amino acids found in nature are of the L type. Hence, eukaryotic proteins are always composed of L amino acids, although D amino acids are found in bacterial cell walls and in some peptide antibiotics. The term amino acids is normally understood to denote the amino alkanoic acids H3N plus Cr1R2N CO2 with N is equals to 1 for the series of alpha amino acids, N is equals to 2 for beta amino acids, etc. The term dihydroamino acids, specifically 2,3 unsaturated or alpha beta unsaturated 2 amino alkanoic acids H3N plus C is equals to Cr1R2 CO2. It should also be noted that the term amino acids involves all structures carrying amine and acid functional groups, including simple aromatic compounds. For example, anthranylic acid and can also encompass other types of acidic functional groups like phosphorus and sulfur oxy acids. The family of boron analogues R3N, BHR1, CO2R2 signifies a dative bond, has recently been opened up through the amalgamation of some examples. It would take only the replacement of the carboxy group in these organoboron amino acids, that is R is equals to R1 is equals to R2 is equals to H by phosphorus or sulfur equivalents to attain an amino acid that contains no carbon. However, unlike the amino acids containing sulfonic and phosphonic acid groupings, it is rare to find naturally occurring examples of organoboron based amino acids. The term peptides is clearly defined and is not surrounded by any form of ambiguity. It covers polymers formed by the condensation of the respective amino and carboxy groups of alpha and beta amino acids. For the structure with M is equals to 2, that is for a dipeptide, up to values of M is equals to 20, that is an icosapeptide, the term oligopeptide is used and a prefix di, tri, tetra, penta, undeca, dodeca, etc. is used to indicate the number of amino acid residues contained in the compound. Isopeptides are isomers in which amide bonds are in attendance that engage the side chain amino group of an alpha diamino acid, example lysine, or of a polyamino acid and or the side chain carboxy group of an alpha amino di or poly acid, example aspartic acid or glutamic acid. One of the simplest examples is the glutathione. Longer polymers with multiple amino acid bonds are termed as polypeptides or proteins, and the term polypeptides is becoming the most frequently used general family name, though proteins remains the preferred term for particular examples of large polypeptides located in precise biological contexts. Nonetheless, the relationship between these terms is a little more debatable, since the changeover from polypeptide to the protein needs a precise definition and explanation. The figure roughly 50 amino acid residues is widely accepted for this. Insulin, a polymer of 51 alpha amino acids, but consisting of 2 cross-linked oligopeptide chains, is on the borderline and has been referred to both as a small protein and as a large polypeptide. Polyalpha amino acids is a better term for peptides formed by the self-condensation of one amino acid. Natural examples exist such as poly-D-glutamic acid, the protein coat of the anthrax spore. There are evidences from erstwhile research that the textile industry witnessed the promise of polyalpha amino acids as synthetic fibers, but the fusion methodology required for the polymerization of amino acids was complex and unprofitable. Polymers of guarded structures made from N-alkyl alpha amino acids have been synthesized as peptide mimetics and have been given the name peptoids. These can be considered as peptides with side chains shifted from carbon to nitrogen. They will therefore have a very different conformational flexibility from that of peptides and will not be viable of hydrogen bonding. This is the simplest portrayal of providing all the correct side chains on a flexible chain of atoms in order to imitate a biologically active peptide. But the imitation can avoid an enzymic breakdown before it reaches the site in the body where it is needed. As per the definitions of polymer chemistry, polypeptides made from 2 or more different alpha amino acids are copolymers or irregular polyamides, whereas polyalpha amino acids are homopolymers that could be described as members of the nylon fraternity. B.Sc peptides are also close associates of peptides with one or more amide bonds substituted by ester bonds. Alternatively, they are formed by condensing alpha amino acids with alpha hydroxy acids in a variety of proportions. There are several important vital natural examples of these of defined sequence. For example, the antibiotic valinomycin and the family of Iniatin antibiotics. Moving ahead, we will now learn principal structure of amino acids and peptides. Amino acids are correlated by peptide bonds to form polypeptide chains. Proteins are defined as linear polymers formed by linking the alpha carboxyl group of one amino acid to the alpha amino group of another amino acid with a peptide or an amide bond. The formation of a dipeptide from two amino acids is accompanied by the dwindling of a water molecule as shown here in the figure. The balance of this reaction lies on the side of hydrolysis rather than synthesis. Hence, there is a requirement of an input of free energy for the biosynthesis of peptide bonds. Nevertheless, peptide bonds are quite stable kinetically. It is observed that the lifetime of a peptide bond in aqueous solution without the presence of a catalyst approaches 1,000 years. A polypeptide chain is nothing but a cluster of amino acids joined by peptide bonds. Each amino acid unit in a polypeptide is called a residue. A polypeptide chain has polarity because its ends are different, with a alpha amino group at one end and a alpha carboxyl group at the other. By principle, the amino end is taken to be the beginning of a polypeptide chain, and so the sequence of amino acids in a polypeptide chain is written starting with the amino terminal residue. Thus, in the pentapeptide, tyrosine glycine glycine phenylalanine leucine, abbreviated as YGGFL, phenylalanine is the amino terminal, that is N terminal residue, and leucine is the carboxyl terminal, that is C terminal residue, as shown here on the screen. Leucine, phenylalanine, glycine glycine tyrosine, that is LFGGY, is a different pentapeptide with different chemical properties. Let us now dwell in the structure of a polypeptide chain. This chain consists of a main chain or the backbone, which is regularly repeating part, and distinctive side chains, which is the variable part. Refer the figure shown here on the screen. The backbone of the polypeptide is effluent in hydrogen bonding potential. Each residue contains a carbonyl group, which is a good hydrogen bond acceptor, and with the exception of proline, a NH group, which is a good hydrogen bond donor. These groups interrelate with each other and with functional groups from side chains to even out particular structures. Proteins are the natural polypeptide chains which contain between 50 and 2000 amino acid residues. Peptides made of fewer numbers of amino acids are called oligopeptides or simply peptides. The mean molecular weight of an amino acid residue is about 110, and so the molecular weights of most proteins between 5500 and 2,20,000. The structures of the peptides are arrived at by a combination of techniques including quantitative hydrolysis and assay of the amino acids present, terminal amino acid analysis, and stepwise removal of amino acids from the amino and carboxyl ends of the peptide chemically or with enzymes, and partial hydrolysis to smaller peptides chemically or with enzymes, which are then analyzed. Properties of amino acid and peptides. Some common characteristics of these amino acids are as mentioned here. All amino acids are 1 degree amines with the exception of proline, and all are chiral with the exception of glycine. The configurations of the chiral amino acids are the same when written as a Fisher projection formula, and this was defined as the L configuration by Fisher. The R substituent in this structure is the remaining structural component that varies from one amino acid to another, and in proline, R is a 3 carbon chain that joins the nitrogen to the alpha carbon in a 5-membered ring. Applying the Kahn-Ingold-Prelog notation, all these natural chiral amino acids with the exception of cysteine have an S configuration. For some amino acids, R substituent is a hydrocarbon and some of them have hydroxyl functional groups. Also, there are variants which have thiol and sulfide groups respectively. Lysine and arginine have basic amine functions in their side chains. Histidine and tryptophan have less basic nitrogen heterocyclic rings as substituents. Finally, carboxylic acid side chains are substituents on aspartic and glutamic acid. Carboxylic acids have pKa's near 4.5 and the conjugate acid of the amine has a pKa of 10. The simplest amino acid alanine has a very high melting with decomposition. It is insoluble in organic solvents and a million times weaker as an acid than ordinary carboxylic acids. The formulas for the amino acids are simple covalent bond representations. Compounds like isobutyric acid, lactic acid, 3-amino-2-butanol and alamine are roughly of the same size and all have moderate to excellent water solubility. Compounds like isobutyric acid and lactic acid are simple carboxylic acids and the compound 3-amino-2-butanol is an amino alcohol. All 3 compounds are soluble in organic solvents, example like ether, and have relatively low melting points. Since amino acids as well as peptides and proteins incorporate both acidic and basic functional groups, the predominant molecular species present in an aqueous solution will depend on the pH of the solution. Furthermore, we discuss nomenclature of amino acid and peptides. The symbol or the denotations chosen for a given amino acid is derived from its trivial name and is usually the first 3 letters of the name. The trivial names of the alpha amino acids that are commonly found in proteins are represented in the genetic code along with their symbols, systematic names and formulas. The general practice is that it is written in capital letter followed by 2 lowercase letters. For instance, uppercase G, small case ln, and not all lowercase, example GLN, or all uppercase, example capital GLN, regardless of its position in the sentence or paragraph. When the symbol is used for a purpose other than representing an amino acid residue, example to designate a genetic factor, 3 lowercase italic letters may be used, example lowercase GLN. Thus, it has to be noted that when the phrase amino acid is a qualified noun, it contains no hyphen. A hyphen is inserted when it becomes an adjective so as to join its components in qualifying another noun, example amino acid sequence. Oftentimes, it is not very easy to use the 3-letter system with long protein names. In order to make the process convenient, a single letter code has been designed which comes to one's rescue in summarizing large amounts of data and for aligning and comparing homologous sequences. In three-dimensional pictures of protein molecule, three-letter code is used. The systematic names and formulas given refer to hypothetical forms in which amino groups are unprotonated and carboxyl groups are undissociated. This convention is useful to avoid various nomenclatural problems, but should not be taken to imply that these structures represent an appreciable fraction of the amino acid molecules. The trivial name refers to the L or D or DL amino acid. For those that are chiral, only the L amino acid is used for protein biosynthesis. Use of the one letter symbols should be used sparsely and should be limited to the comparison of long sequences. The fully systematic forms ethanoic, propanoic, butanoic and pentanoic may alternatively be referred to as acetic, propionic, butyric and valeric respectively. Similarly, butanedioic is succinic, 3-carbamoyl propanoic is succinamic, pentanedioic is glutaric and 4-carbamoyl butanoic is glutaramic. The symbol uppercase A, small case Sx denotes aspartic acid or aspargine. Likewise, uppercase B denotes uppercase N or uppercase D. Uppercase G, small case Lx and uppercase Z likewise represent glutamic acid or glutamine or a substance such as 4-carboxyglutamic acid. Uppercase G, small case La or 5-oxoproline, uppercase G, small case Lp that capitulates glutamic acid on acid hydrolysis of peptides. Now, let us throw some light on the naming convention used while naming peptides. The names of acyl groups ending in YL are used. Therefore, if glycine and alanine were to condense so that glycine acylates alanine, the dipeptide formed is named glycyl alanine. While a condensation in the reverse order will name the product as alanyl glycine, the name of the peptide begins with the name of the acyl group representing the N-terminal residue, and this is followed in order by the names of the acyl groups representing the internal residues. Only the C-terminal residue is represented by the name of the amino acid, and this closes the name of the peptide. The sequences of higher oligopeptides and polypeptides of biological origin are usually described more conveniently by symbols rather than by constructing lengthy names, and they often have trivial names. Lastly, in this session, we will talk about difference between amino acid, peptides, polypeptides and proteins. Amino acids are the basic units which form the foundation of peptides, polypeptides and proteins. Therefore, the 3 P's, that is the peptides, polypeptides and proteins, are built from amino acids. The basic difference is the number of amino acids which constitutes them. One can arbitrarily state that a molecule with 50 amino acid units or less is considered a peptide and one with more a polypeptide. Proteins are made up of one or sometimes more than one polypeptide linked together. For instance, pepsine is made of one polypeptide with around 320 amino acids, but hemoglobin constitutes of 4 polypeptides with around 140 amino acids in each. An amino acid is one unit of a peptide chain. When 2 amino acids connect, they form a peptide bond. When you have lots of amino acids connected together in a line, they are called a polypeptide. Thus, a protein is made up by one or more polypeptides and usually has hundreds if not more amino acids in them. At the end, we conclude the session. L-alpha amino acids are present in proteins, are the principal constituents of proteins, and both D-amino acids and non-alpha amino acids occur in nature. The basic elements of all amino acids are at least two acidic functional groups, RNH3 plus and RCOOH. Many also possess additional weakly acidic functional groups such as OH, SH, guanidino or imidazole moieties. The pKa values of all functional groups of an amino acid dictate its net charge at a given pH. Pi is the pH at which an amino acid bears no net charge and thus does not move in a direct current electrical field. Of the biochemical reactions of amino acids, the most important is the formation of peptide bonds. The R groups of amino acids determine their unique biochemical functions. Amino acids are classified as basic, acidic, aromatic, aliphatic or sulfur containing based on the properties of their R groups. Peptides are named for the number of amino acid residues present and as derivatives of the carboxyl terminal residue. The primary structure of a peptide is its amino acid sequence starting from the amino terminal residue. The partial double bond character of the bond that links the carbonyl carbon and the nitrogen of a peptide deliver 4 atoms of the peptide bond coplanar and limit the number of possible peptide conformations. The structure of a peptide can be written comparatively easily without showing the complete amide synthesis reaction by learning the structure of the backbone for peptides and proteins. Thank you.